we have 1 minute. Holy moly.
This is good. This is what you want.
Yeah. There's 2 seats up here. Cool. Yeah, Sandy and Rick texted me earlier, so. Hey, how's it going? You're so close. I might like trip and fall on you. This is going to be, yeah. Yeah.
All right. It's 4 o'clock and it looks like everybody is settled in. Thank you so much for showing up to this special time seminar. It is my great pleasure to introduce to you Ben Perlman, who has been a graduate student in my lab the last 5 years. Ben got his bachelor's at Uncle Charlie's summer camp, also known as University of California, Santa Cruz, which happens to be my husband's alma mater, which is kind of weird. And then he did his master's degree with one of my lab mates from graduate school, Laura Ferry, at Moss Landing Marine Lab. And then he came to us, and most of you know Ben pretty well. He's made a mark in the department. Not only has he been incredibly enthusiastic getting his own research pursued, but he's directed numerous undergraduates. He's helped develop an entire new course centered around Belize, which has been wonderfully successful. And I'm, kind of thinking, is there some way that I can flunk him so that I can flip him around right now? But, I guess not. So we're going to have to let him tell you about his dissertation research, biomechanics of an odd little fish that spends time on land, kinematics, kinetics, and motor control of the mangrove rivulus. Take it away,
cool.
Ben.
All right, thank you so much for coming. It's nice to see all your faces, especially a lot of people from my spring Belize class just trickling out of the woodworks. If you don't mind turning all the lights off, hopefully nobody will fall asleep. I am trying to convince you that this fish is worthy of your time and darkness. So let's go ahead and get started. So it is a central tenet in comparative functional morphology that form follows function. So to elicit our first example, we have this beautiful albatross with these long, slender wings, which allow it to glide over entire ocean basins. We also have the cheetah, which is able to stealthily chase potential prey items through the open savanna with the ability via its extendable hind limbs. It has a very flexible vertebral column and a very streamlined body profile. We also have tuna that also have a streamlined body shape along with this deeply lunate tail, allowing it to cruise through pelagic waters. The arboreal nature of this chameleon is possible because of its modified forelimb and hind limbs, allowing it to easily grasp tree limbs and branches as it moves throughout the forest. This kangaroo, which is a saltatorial jumping, hopping champion, is possible based on the spring-like tendons in its hind limb, allowing it to move very efficiently through these open grasslands. So in these 5 examples, these animals are specialized to move in a particular environment. Now, what happens if an animal occupies 2 extremely distinct environments, such as the mud skipper, an amphibious fish? So they have modified pectoral fins, which are extremely strong, stout appendages, allowing it to crutch along these muddy substrates. So we have this expectation that anatomical specialization will provide evidence for what the animal actually does. So we see this expectation in this artist's rendition of these early tetrapods with these stubby little pectoral fins which evolved in the water, which eventually gave rise to limbs for terrestrial locomotion. Now, these major functional transitions are often accompanied by morphological changes. So here we have Tiktaalik rosier. It's an early tetrapodomorph fish from the late Devonian, about 365 million years ago. Now we have this fossil example on the left with the artist's reconstruction on the right, and I want to point out their pectoral fins, which have been modified along with fin rays that have bendable elbows, a very flexible wrist, allowing them to prop themselves off the ground. They also have a neck joint, which allows the head to move independently of the body for more efficient locomotion out of the water. So what happens if these morphological, these functional transitions are not accompanied by a morphological change? So if you take this fish, for example, the mangrove rivulus, Kryptolebias marmoratus, and throughout the rest of the talk I'll most likely refer to this as a KMAR for short, would you be able to suspect that this fish was anything but fully aquatic based on its external morphology? So what's really interesting about this particular fish is its ability to move on land. So this is one of the first videos ever captured of these fish, and they're actually jumping out of a log. Now, what I want you to really focus on here is the fact that they are directing their movement away from this person's hand to efficiently get away and escape this potential predator. So let's learn a little bit more about the background of this fish. They are a quasi-amphibious teleost, a bony fish that makes occasional forays onto land. They're found from central Florida down to southern Brazil in the Atlantic, along with the Caribbean islands. Now, this is the general orientation the fish takes in, that it occupies in water. So unlike the late Devonian Tiktaalik example, which has obvious external morphological features, this fish lacks any obvious external morphological characteristic, suggesting that it would be able to efficiently move from water to land, let alone coordinate its behavior out of the water. So let's focus on some of its fins. They have a streamlined body shape, and here's its dorsal fin, which is set far back along the body, directly opposite its anal fin. They have a nice lobed plate-like tail fin, the caudal fin. They have their plate-like pectoral fins up by the cranium region. Now, this picture on the bottom right is a fish in its general orientation when it's out of the water. We took this in the field. Here's its head over on the left, and its tail curved on the right, sitting on this nice little leaf. So this species has become an important model organism, mostly because of its reproductive strategies. They are one of only 2 known vertebrates to science to be a self-fertilizing hermaphrodite. So they're simultaneously a male and a female via their ovotestis. They have sexual fertilization, which occurs internally. They lay their eggs externally via oviparity. Their offspring are technically isogenic clones because they share the same genetic material as their parent. And one thing that's really kind of bizarre, they are also males. There's no females known in their population, so if the eggs are raised in cold temperatures, they will be born a male. They could be born a hermaphrodite, and then they could basically stay in cold water for an extended period of time and then shift to a male, which is known as a secondary male. And they are not known to shift back to a hermaphrodite. So each individual will lay an egg and can eventually, via multiple generations, give rise to an entire population that you see here. Some of the reasons these fish leave their aquatic habitat are due to poor water quality. They could be searching out a prey item such as other fish, mosquito larvae, and insects like crickets and spiders. They're also known to escape predators such as snakes, birds, and fishes. This KMAR over here didn't get the memo because this mangrove water snake, Nerodia clarkii, doesn't really seem to care about its prey item right here, and he doesn't seem to be very afraid of this particular snake, which is really weird. The snake eventually ate that fish. So one of their interesting adaptations for remaining out of water, and there was a study done by Scott Taylor in 1990 that recorded these fish to remain out of the water for 66 days, so just over 2 months. Now here is a picture of their gill structure. In the water, you'll see their secondary lamellae with all this space in between. Now, when they've been immersed out of the water for 24 hours, you can see that the interlamellar cell mass has completely filled in. So they're able to reconstruct their gill filaments to decrease the opportunity of desiccation and collapse of their gill structures. So We know a little bit about their life history and their physiology, but we still don't really understand anything about how they generate these terrestrial locomotor behaviors. So we explored several aspects of how they do this because we want to understand how these fish are actually accomplishing the feat of terrestrial locomotion. So the first two questions, how does this fish transition from water to land? And once it's on land, how are they capable of locomoting there? Are mostly exploratory studies. We then have some hypothesis-driven experiments looking at the forces they generate when moving on land, followed by the motor pattern that they use to drive these behaviors via their axial muscles when they're out of the water. So the first thing we have to do is collect our specimens. So we go into the mangrove swamps. I am completely covered. It was 103 degrees that day, 90-something% humidity, and you don't want to get mosquito bitten. So my hands were eventually covered up, but it's hot and crazy, and that's what you look like, but it's worth it because you get fish. So we go to our little Kmart and get our animals. So on the top left, so Miami is right up here for reference. These are the Florida Keys. So here is Key Largo. We go to a slew of places along the Keys. And then my favorite place in the world, here is Lighthouse Reef Atoll. It's about 50 miles off the coast of Belize City in the Yucatan. And to blow this up, this is about 20 miles the way the crow flies. And we sample in the mangrove keys, such as Long Key in the south, and Northern and Sandbar Key in the north. And if you kind of squint and use your imagination, I think Lighthouse Reef looks just like a Kmart. So that was very convenient. So these dots in red denote the locations where we collected all these fish from multiple populations genetically and geographically distinct. So here is what our field site looks like on Long Key. The two red circles denote some of our sites. This is also the base of the operations for our Lighthouse Reef class. We stay right here and snorkel all these places throughout the atoll. So here is a picture of a drone that Max Messenger, a grad student with Miles Silman, was flying at about 1,000 feet over us, while myself, Dr. Miriam Ashley-Ross, and a slew of students from a year ago were sampling in the mangroves. Let's take a dive into the canopy, under the canopy. This is what the red mangrove swamp looks like, species Rhizophora mangle. So these fish are occupying these watery habitats, and they have to negotiate around all of these prop roots. And pockmarked throughout these prop roots are a bunch of crab burrows. So on the left, here's the opening to a crab burrow occupied by a land crab, Cardosoma guanhumi. It's about the diameter of a softball. Now on the bottom right are the cup traps we use, and you can see them outlined in these red circles of how we capture the fish. So no, I didn't steal this from the pit. That's the cafeteria at Wake Forest, if you're unfamiliar. We actually ordered these. They weren't repurposed. We take a little funnel, insert it into the cup, little 16 ounces, and we drill holes in this, and then we stick this into the crab burrow or in between a bunch of prop roots, wait for 1 to 2 hours, and allow the fish to hopefully swim in, not swim out, and we'll get anywhere from 1 to 6 fish at any given time. You'll notice there's no water. The tide has gone out. And this is the optimal time to capture these particular fish. So I'll just pass this around if you want to see. All right, so you have to be very careful where you place your prop root because saltwater crocodiles occupy a lot of these areas. Miriam and I luckily still have our hands, and we're not the prey consumed by this little guy. We wait an hour and a half, and we get to explore wonderful places such as this while we are waiting for fish to occupy these cup traps. We then revisit the cup traps, and the first thing we do after we collect them, hopefully, is take a little fin clip from their tail fin, and we send that to a colleague of ours, and we genotype every single individual. So you can't actually see it, but I'm manipulating the fish, and a bunch of students, some of whom are in the audience, were assisting. Here you can see some of the cup traps for size. So we then put them in little whirl packs. It's basically a glorified Ziploc, and we line them up with about 30 mils of water and take them back to the States in a nice inconspicuous carry-on bag. So we're now back in the lab. Here is my colony at Wake Forest, and there is one fish occupying each 32-ounce container. They're in brackish water. It's temperature controlled and everything like that. They're all marked, and we then go to work. So to remind you what the first question was, we want to explore how these fish transition from water to land. So through a series of natural history papers, we know that these fish actually pursue prey items out of the water. So we were able to train these fish to capture this pinhead cricket on a modeling clay platform. So the fish, what you're going to see in the successive video, is moving from the left to the right. It's usually not this wavy. We have two high-speed video cameras, one in the dorsolateral position and one in the lateral position, which is where your eyes are currently in. So you're going to watch this fish launch out of the water from left to right, and you can see that he overshoots the clay platform and falls into the water on the other side. This behavior occurs at about 27 body lengths per second. Now, that's incredible because if you are familiar with salmon, they leap out of the water at about 10 body lengths per second. So these fish are performing at 2.5 times that rate, which is amazing. They have a takeoff angle of approximately 47 degrees. So to have maximum horizontal displacement, you would predict 45 degrees is what you would achieve, and that's exactly what we see with these fish on average. They also reach a height of about 3 to 4 body lengths. So from the aerial position, you'll see how this fish actually generates this particular behavior. So in the still image sequence, starting on the top left, they make an S-bend with their body. So they bring their tail up toward the cranial region, and they accelerate out of the water. And this all occurs in about 50 milliseconds, and that is the launch behavior. This occurs about 55% of the time of their three major behaviors. So the second most common behavior is the pounce, which is basically a truncated launch. So you'll notice that there was a pinhead cricket at the water-land interface. It made purchase with the item, immediately takes it into the water, violently thrashes it around to force it down its buccal cavity for eventual consumption. And this behavior only occurs when the animal is feeding. So the third most common behavior is the squiggle. This is published. It is now a scientific term. Use it as much as you want. It's basically like an army crawl. So you can see that the pectoral fins, the side fins, are making purchase with the substrate. They will use that to pivot around with their body as their fulcrum as they then continue to move forward from water onto land. And you can see there is a lot of swinging going on with their tail, where about 20% below the head is staying somewhat still through that entire process. So here are some data. There's a lot to walk you through, so let's go. So here we have this nice little silhouette. This black symbol denotes the head, followed by the red triangle, all the way down to this magenta X representing the tail. Now the axes are the absolute space in which the body parts are moving forward. So let's start with the head region. So this open symbol represents the first position of movement. You can see that there's lots of lateral undulation with the head. You can see right here 20% of the way through the body, which is close to its center of mass. You can see very little lateral movement. It's staying mostly still as the head swings side to side. Starting with the tail over here, you can see it makes this figure 8 type movement. And that's reminiscent of both aquatic and terrestrial fishes, such as eels and ropefish. It's a pretty common behavior. And we see that with the caimar. So basically the take-home is lots of movement with the head and the tail, and the mid-body is staying mostly still as it squiggles forward. So you see this starting here, moving toward the top of the slide. That's because everything is moving forward. Now here we're measuring amplitude. So this 0 axis corresponds to this horizontal orientation of the body. The symbols represent the same thing as the previous slide. So when the fish's body moves toward the left, that we just denote is a positive. And when the fish's body moves toward the right, we have that as a negative. So on the bottom, you can see through an entire cycle, wave cycle of the squiggle, there's a lot of undulation, again, with the head in black, and the tail, you can see, also has a lot of undulation. And at 20%, you can see, again, it's staying very, very consistent along this horizontal 0 axis. So not much movement with the mid-body, lots of undulation with the head and the tail as it squiggles forward. So there were statistical differences between the head and tail regions and the rest of the body in terms of amplitude. We also measured the distance ratio, which is just the linear forward displacement divided by the absolute lateral undulation. So the ratio, if you have a small ratio, it's because you have a lot of lateral displacement. If you have a really large ratio, it's because your body was mostly profile without moving side to side. Again, we have the head on the left and the tail on the far right. Those were significantly different from the mid-body, which again had greater ratios representing lower lateral displacement. So now we know that these fish use a launch, a squiggle, and a pounce to move from water to land. So how is it that these fish are actually locomoting on land once they reach that particular area? So we use high-speed videos to record the actual behaviors elicited by these fish, and we have some kiddie pool trials with a Kodak PlaySport non-high-speed video to record a bunch of behaviors from a bunch of populations from Florida and Belize, and the Bahamas and Honduras and some other locations which we also were able to select. So this is the tail flip behavior, and this will play a few times. Notice that I use a metal rod to elicit this particular tail flip, and they are extremely acrobatic characters. So they will lift their head off the substrate, generate a C-bend, which is reaching maximum body curvature, which we call stage 1. They will eventually continue through stage 1, that maximum C-bend, which you'll see in just a second, and they will launch off the substrate, becoming a ballistic object. That denotes stage 2. So that whole behavior is a tail flip. They are able to move anywhere from 5 to 10 body lengths. That's like you or myself jumping 60 feet from a standstill. They're extremely acrobatic. They will rotate anywhere from 1 to 5 times in the air. And they always have their tail pointing in the direction in which they will land, and their head is always facing their perceived threat. And we see this time and time again. We're almost ready to call this a stereotyped behavior, but there's a few other things to work out before we do just that. So what you saw before, the fish was on its side, its lateral aspect. In this particular orientation, the fish is on its ventral surface in the prone position. So here in the illustration on the bottom left, just to remind you, this is the dorsal fin, here's their tail fin, and then here we have their anal fin. So when they're on land, sometimes they will torque their body in this axial twisting behavior about 90 degrees. So what is very interesting to take away, here's their pectoral fin, here's their dorsal fin, here's their anal fin, and these median fins are now acting as paired fins, which most likely provide additional stability when these fish are on land. Also notice that their tail fin is now rotated completely flush with the substrate. So since they lift their head up and away from the perceived threat, and they will be jumping in the direction where the tail is pointing, they now can provide increased thrust with this greater surface area as they launch off the substrate. So here's a still image sequence of that tail flip. Again, stage 1 is reaching maximum body curvature. Stage 2 is when the fish has launched off the ground. We also see a reorientation behavior. So this asterisk denotes the location of the perceived threat. The arrows on the bottom are representative of the head position of the animal. So when this fish tail flips some distance away from its initial location, its head might not actually be facing the direction in which it came. So it will then squiggle and reorient its body to face that particular direction, as you see here on the right, to then set its body up for another series of tail flips away from that potential threat. And we also see the squiggle behavior as well on land. So we have the tail flip, these reorientations, and the squiggles. So now we understand how they locomote on land. For the rest of the talk, I'm going to be focusing on the tail flip because of its unusual behaviors. And let's start with some of the forces they generate when moving on land. So I'm going to be comparing these adult mangrove rivulus to exclusively aquatic juvenile largemouth bass, Micropterus salmoides, as you see here. Some of you may have fished for them. And we basically want to figure out who's the Olympic champ in the forces generated based on their two ultimately very distinct behaviors. So this is me. Here is a nice kiddie pool that's empty with wetted bench liner paper. There's the fish. I'm going to use my finger of doom to chase this fish across this shallow, empty arena. And you'll notice that, let's play this again, it is tail flipping and then reorienting its body as it moves across and away from my finger. So here's the path that it takes, this red trackway, and I'll be using red to denote the Kmar for the rest of this section, and blue will represent the bass. You'll see that coming up. So we have this coordinated tail flip behavior on land, which is nicely contrasted by an uncoordinated thrashing behavior that we see all the time with these juvenile largemouth bass. You'll notice that there's absolutely no directional component. So the Kmars have a lot of lateral displacement, where these bass have very erratic to little displacement. And that trackway looks something like this. Now, to compare with the Kmar, we started to predict a bunch of hypotheses that there's something going on with the forces in terms of their directionality based on a coordinated versus uncoordinated fish behavior. So let's start with the mangrove rivulus. So we predicted that these ground reaction forces would be greatest in the antero-posterior direction, that's the forward-back direction, based on their directional displacement. We also predicted that there would be a longer duration to reach stage 1, the maximum body curvature, and the time it takes to launch off the plate. So you can see the fish in its maximum body curvature through the tail flip. We also predicted there would be a greater impulse, which is the force generated multiplied by the duration spent on the force plate in the antero-posterior direction. So we have time on the x-axis, force on the y, and we standardized everything in body weight. It's just newtons per kilogram. We did this to standardize so we could compare across species and size of these two fishes. So for the antero-posterior direction, we predict the Kmar to have a force profile like this, with bass in blue much lower, based on their little to erratic displacement. So with the bass, we predicted their ground reaction forces to be mostly in the vertical direction based on almost no displacement, and a shorter duration to reach maximum body curvature and launching off the plate. So this is what the fish look like in the max body curvature. And a greater impulse, again, in the vertical direction, because we predict that there's little to no lateral displacement. And this is what you would see with the Kmar, and a greater vertical ground reaction force in the bass in blue. This is what our experimental setup looked like. We had a multi-axis fort platform that we recorded 3 directions. So we have the vertical going up, we have the antero-posterior horizontal, and the medial-lateral side to side. So we place the fish here and we record the ground reaction forces. We use 2 high-speed video cameras at 500 frames per second, and this mirror at a 45-degree angle to capture the aerial position. So here's what the mangrove rivulus looks like. Notice that the fish is tail flipping from the prone position. So we have the lateral view on the left, the dorsal view on the right, and you've seen this tail flip behavior. Now we have the thrash up for the bass as it lifts its head off the substrate, reaching maximum body curvature. And again, pretty obvious compared to the Kmar, little to no displacement. We also observed what we denote as a thrash down. So using the same exact stimulus, it was unpredictable whether the fish would lift its head up or drive it into the substrate. So nevertheless, you see a very similar behavior. Again, there was some lateral displacement, but very minimal compared to the Kmar. So we subset the data to compare the thrash up and thrash down compared to the tail flips that we see in the Kmar. So here are the first data slides. So on the x-axis, we have jump percentage. So we have 0, which is where the first movement began. 100% of the jump is when the fish was completely off the plate. All the Kmars are in this reddish color, and the bass, again, are in blue. These solid lines represent the mean, with the shaded areas being the standard error. We used a student's t-test to compare the bass to the Kmar. So you can see there was no statistical difference between the vertical ground reaction forces in the bass and the Kmar, as their peaks are up here. In the antero-posterior direction, again, it was not statistically different. You'll see the bass at about 0.5 with the Kmar at about 0.3. The reason you see this in negative direction is because it's denoting the direction in which the fish jumped off the plate. Now in the medial-lateral direction, again, not statistically different. Here you can see the bass up here and the Kmar down there. Now, what was really interesting, we have the peak net ground reaction force. So the peaks, again, were not statistically different, but what was the time it took to reach the peak ground reaction force. So the bass reached their ground reaction forces at about 60% of the way through the jump, where the Kmars reached their ground reaction forces at about 85% of the jump. So even though the forces were not different, you can see here in vertical they were about the same with anteroposterior and medial lateral. So like everything is in life, timing is everything. These fish are operating on two extremely different time scales, and that became extremely important in how these particular behaviors are generated. So it wasn't just the magnitude of the forces, it was the actual time it took to generate these particular behaviors. So we see that really nicely with the total duration. Stage 1 is in white, stage 2 is in gray, and you can see that the tail flip was much longer. This was just statistically different than the bass when they do a thrash up versus thrash down. Now, what I have in the horizontal solid white, this is the aquatic stage 1. So when fish escape and generate that max body curvature in water, that occurs in about 30 milliseconds. And you can see that both the Kmar and the bass exceeded that time. This horizontal dashed line represents the aquatic fast start, which by definition occurs in 100 milliseconds. You can see that the Kmar exceeded that, whereas the bass, it was well within stage 1 and stage 2 of that fast response. Nevertheless, the duration being much longer in the Kmar compared to the bass. Now, when we pile in the forces along with the duration, reminding you that impulse is the time it took to move itself off the plate multiplied by the ground reaction forces generated in that behavior, we have the impulse. So we have the same pattern actually in the Kmar and the tail and the bass. So the vertical impulse was greatest, followed by the mediolateral in black, with the anteroposterior impulse being the lowest for the Kmar and the bass in both of those behavioral modes. Now, it's kind of bizarre to think that the impulse was so much greater in the vertical compared to the anteroposterior for the Kmar, since they generate these lateral displacements. So we still see the anteroposterior, that front-back impulse, greater than the bass. But like frogs, which generate most of their ground reaction forces in the vertical direction, and they're still capable of launching themselves 10 plus body lengths away from their initial location, we see a similar pattern with the Kmar. Now, as predicted, their lateral displacement for the Kmar was about 180 degrees, where the bass was about 3. So what does that look like? When the fish start, here's their head position for the Kmar on the right, along with the bass. When they completed their behavior, the head location was also in the same orientation for the bass and about 180 degrees away for the Kmar. So as expected, and we see that very nicely here. So to summarize the ground reaction forces, so the kinetic data for the mangrove rivulus, we noted that the forces were similar in the rivulus and the bass. We saw these differences in the timing of peak ground reaction force. And the impulses were greatest in the mangrove rivulus for all 3 planes compared to the bass. Now, kinematically, there was a longer duration in the mangrove rivulus to reach maximum body curvature and to launch off the plate compared to the bass. And we saw greater horizontal displacement in the mangrove rivulus compared to the bass, again, as expected. So now we wanted to figure out what's going on with the neuromuscular system in terms of the motor control. What's this motor pattern in the axial muscles when these fish move on land? So here is what the aquatic sea star escape response looks like. So here's this fish at rest. The asterisk denotes where this perceived threat is going to come from. The fish will reach a maximum body curvature, the sea bend, hence the name sea start escape response. And the completion of that is stage 1, very similar to how we defined it in the tail flip. And the fish will then swing its body, its tail, away from the body to propel itself away from the perceived threat. That will be the completion of stage 2. Again, this entire behavior occurs in about 100 milliseconds. So here is an electromyogram recording the muscle activity of the fish. So this happens to be a rainbow trout, Oncorhynchus micus. So these first 3 panels are on one side of the body. Now at stage 1, again reaching that maximum body curvature, you'll notice there's muscle activity on one side of the body and no activity on the side. However, in stage 2, the opposite side of the body will activate its musculature to swing its tail around its body and propel itself through the end of stage 2. So keep that in mind because you're going to see some electromyograms in a few slides. So this whole escape response is driven by a series of escape neuronal networks, mostly the Mothner neurons, which are a pair of bilateral neurons in the hindbrain of a fish. So this side is the rostral part of the fish. Here's the caudal. So here's these two Mothner neurons, and their axons transcend to the opposite side of the body. So depending on where the stimulus is coming from, the fish will either turn to one side versus the other. So here again is a diagram of the tail flip. This asterisk denotes where that perceived threat is coming from, and it superficially looks like the aquatic escape response, hence naming it stage 1 and stage 2. So this raised the question, do mangrove rivulus use similar motor control for both the aquatic escape response and their terrestrial tail flips? So we collected a bunch of KMARs, and we inserted 5 fine wire bipolar electrodes percutaneously into repeatable locations of these fish. Those locations are denoted by these orange dots. So we have paired left and right side of the anterior epaxial muscles, and we have paired left and right side posterior electrodes in the posterior epaxial muscles. Again, this asterisk denotes where the threat is coming from. The fish will curve into this C shape. When it does so, the white denotes the ipsilateral muscles, and that's with respect to the maximum curvature of the body. The yellow will represent the contralateral muscles with respect to the curvature of the body. Here we have the lateral view. So this horizontal line is the horizontal myoseptum with the epaxial musculature above that. So here's the paired anterior, and here's the paired posterior muscle electrodes. We also had a 5th electrode into the body cavity to act as a ground. Now, these fish are about 0.6 grams in mass. So here is a US penny. For reference, these fish weigh about 25% of that penny. So to our knowledge, this was the first successful attempt at getting electromyographic results from a vertebrate. So very, very cool. Here's what our experimental setup looked like. So we put these fish in a 10-gallon tank. Here's the electrodes connected to a breakout box, then connected to an amplifier to amplify the recordings, and we see that on a computer. We have this Faraday cage to remove noise. We immediately, after recording the electromyograms from the aquatic stage, we put it on this wetted bench liner to record the tail flip behavior successively. And here we had some LEDs and a high-speed video camera in the lateral position. The variables we measured were the onset time to muscle activity, the duration of those muscles in the aquatic escape response and the terrestrial tail flip, and the rectified integrated area of the muscle burst, which is basically the intensity of the muscles. We put this into a multiple analysis of variance, and we had media as our fixed factor. So here we have the end of a dip net. To elicit this aquatic escape response, you're going to see the fish turn away from the dip net. So there it reached its maximum C bend and eventually propelled out of that. And this we then followed by the tail flip. Notice we used 2 different types of stimuli. We had the dip net and my finger. There were no statistical differences using various predators, if you will. And you'll see that it raised its body off the substrate, reached the maximum C bend, and landed some distance away from its initial location. So here is our representative electromyogram. On the left column, we have the aquatic sea start. On the right, we have the terrestrial tail flip. So again, everything in white, those are the ipsilateral muscles with respect to the body curvature, and again, the yellow being the contralateral. So in stage 1 of the aquatic escape response, everything in light gray, the width represents the duration of stage 1. You can see it was much shorter in water compared to land, and these diagrams represent the body position of the fish at the end of stage 1, again reaching max body curvature. So as expected, we see synchronous muscle activity in stage 1 aquatically, and this was actually asynchronous statistically, which is what we would expect as we see in other exclusively aquatic fishes. However, terrestrially, we see this asynchrony in stage 1. And what we think is going on is the fact that these fish are preparing the muscles to then enter through the end of stage 1, that max body curvature, press down on the ground with their caudal peduncle, and launch off the substrate. So that takes us to stage 2. We see an asynchrony in the contralateral muscles with the aquatic escape, but we see this synchronous muscle activity in the terrestrial tail flip, again driving the fish off the terrestrial ground. So what you're seeing here are the average data of all those trials. So everything in black, that represents the aquatic escape response. Everything in white are the terrestrial muscles, or the muscles in the terrestrial trials. And again, the same as before, stage 1 is the light gray, stage 2 is the dark gray. And what you can see here, the much wider muscle, the height of all of these muscles are much greater on land compared to in water, and that's because these muscles are activated for a longer period of time, because it's a much longer behavior to generate that tail flip compared to the aquatic escape response. So to sort of quickly summarize the motor pattern study, we found this delayed onset time in the aquatic situation. There was longer muscle duration and activation in both stages 1 and 2 terrestrially. There was the concurrent onset of muscle activation in stage 2 on land. There was a greater muscle intensity on land. And we saw this asynchrony in the ipsilateral muscle bursts terrestrially, most likely being responsible for driving that caudal peduncle into the ground to then launch off the substrate. And this extended contralateral muscle activity in stage 1. So based on all of this, it led us to conclude that there is indeed a different motor pattern on land compared to the water. So we're going to be conducting some studies in the future in terms of how do we really separate this different motor pattern, because the media, water versus land, are extremely different, and there's a lot of physical variables you need to account for. So to summarize everything, starting with that water-to-land transition study, we found out that these fish have launches, squiggles, and pounces. Once they're on land, we noted that they tail flip, they produce these reorientations, and we also see them squiggle. The forces generated, the time to peak ground reaction force occur about 85% of the way through the jump for the Kmar, as opposed to about 60% for the bass. The greatest ground reaction forces were in the vertical direction for both species. However, we still saw the greatest impulse in the anteroposterior direction for the Kmar as expected, compared to the bass. And in the motor pattern, as I just said, there was a different motor pattern driving the tail flip compared to the aquatic escape response. Now, what I didn't show was the fact that the bass had the same motor pattern in both water and on land. I didn't show that here, but that, again, is an exclusively aquatic fish, and we do see these differences in motor patterns for these amphibious species. So here is a phylogenetic tree of the actinopterygian fishes. Those are the ray-finned fishes. They represent about 99% of extant living fish. And a synapomorphy for all these fish are these Mothner neurons, which seem to be somewhat responsible, along with a series of other neuronal components, to drive that escape response. So what you see here, the atherinomorphs, the rivulus, mangrove rivulus, we know that these fish have a different motor pattern. We also know that they're capable of tail flipping, and here we have the bass, which is a perciform fish. And then what you see here in yellow are a bunch of other fishes that are also capable of these tail flipping behaviors. Yet we don't know anything about their motor patterns, the kinetics they generate, and it would be interesting to understand how conserved or widespread is this motor pattern in the tail flipping behavior found throughout these actinopterygian fishes. So if we were to experience a situation like this in my artistic rendering of the fossil record, would we be able to predict that the fish on the top is extremely efficient at moving from water to land, let alone coordinating its terrestrial locomotion, compared to this fish on the bottom, which has extremely feeble attempts at moving out of the water? So it basically allows us to somewhat revisit the fossil record to see if indeed form is always following this functional paradigm. So to basically paint a more complete picture of all these things we found out about the mangrove rivulus, I had a slew of undergrads help me with a bunch of side projects to try and figure out what's really going on from a lot of different scientific aspects. So I had Mike Fasano help me looking at the muscle morphology and describing the fiber types that we see in these particular fish. Mark Sims helped me with the navigational ability of these particular fish. Are they able to seek out shelter, different colored objects? And that led to a deeper study of trying to figure out what's going on with the retinal cells, so the eye morphology. Do they have some type of hybrid eye, or are they exclusively aquatic looking, or something that we would expect for something that has terrestrial bouts? We've looked at the ontogenetic effects of the ossification of the vertebral column and the fin rays. Matt Teball helped with that. And we've also begun to explore the mechanics of the vertebral column twisting. There's a few distantly related groups of fishes that are also capable of twisting, such as the Pacific leaping blenny, and we want to know how many vertebrae are involved in that torquing of the body, because it's really bizarre to keep your body in that twisted position for extended periods of time. You're generating a lot of strain on your muscles, so what's going on there? We've also looked at the kinematics across their entire range, from Florida down to Brazil, and those studies are still ongoing. So there's a lot to learn about these particular fish. So with that, I would like to acknowledge a lot of people to help complete this dissertation. First and foremost, Dr. Miriam Ashley Ross, who's been instrumental in allowing me to bug her unscheduled in her office, asking lots of questions. I have things coming up a mile a minute, and she's been more than receptive and wonderful and supportive in every single way. It's been an absolute treat to work with her the past 5 years. I can equally say the same with Dr. Miles Silman, Bill Connor, Wayne Silver, John Long, who came here from Vassar, my outside committee member. You guys have been wonderful in reading through manuscripts, providing terrific ideas, and trying to narrow my focus a bit, as I seem to have a lot of ideas, and it's nice to make sure things are carried to completion before you move on to the next thing. So thank you all. Of course, my lab mates Morgan Burnett and Christine Vega for listening to me give practice talks and talking out scientific ideas, being friends. It's been absolutely terrific. Dr. Ryan Early, who helped me get involved with these fish. He's a colleague of mine from University of Alabama. He invited me into the field to collect these fish in the Florida Keys many, many years ago, and it's been terrific ever since. Dr. Rick Blobe and Dr. Sandy Kawano at Clemson and now Nimbios provided force plate access. Alice Gibb, a colleague of ours at Northern Arizona. Patricia Hernandez at George Washington taught me how to do immunohistochemistry for some of the muscle studies. There's a lot of people. I definitely want to highlight Blake Ross and Sarah Edwards. They own Itza Lodge at Lighthouse Reef Atoll, Belize, and continue to invite us back so we can conduct field studies, among other things. Dr. Dimmick and Jim Curran for inviting me to Belize 4 years ago. So I'm glad that seemed to have worked out well. Dr. Farbach, Len Mars, and Wayne Pratt for letting me use the cryostat. Of course, Cindy and Nancy, the secretaries in the department, for organizing all kinds of things throughout the years. Nick Ristoff and Isaac Banks for providing that absolutely wonderful video at 1,200 frames per second for a nice, crisp, BBC-worthy, I hope, video. Of course, I couldn't do this without my students, Team KMAR, and I guess I'll thank the BASS people, but a slew of undergrads who did research fellowships with me and have analyzed some data, and just talking science. It's been great. Funding sources and permitting, things like that. I especially want to give a shout out to Bio 311, 312 students, and of course Miles and Miriam for making one of my goals become a reality. We've now done this 3 years, and hopefully it will continue into the far future when I'm gone. Please let me come back in some form. This has been great, and it's also allowed us to spread the love of KMARs and mangroves in particular. So without further ado, I'd be more than happy to entertain any questions.
Somebody hit the lights. Questions for Ben?
tested on this then?
Okay. Are there females in the population?
No.
Okay, you were listening. Good job. Yeah, Jason.
If this is an escape response, why are they jumping in different holes?
So that's a really good question. There's no detriment by living among these crabs. They don't feed on them or anything like that. In fact, it's thought that the fish are sometimes picking off some of the biofilms from the crabs and also maintaining the burrow, because the fish also eat a lot of the mosquito larvae that you see in the crab burrows, if that has any effect. But Mike?
Two unrelated questions. How does the weight distribution or mass distribution of the juvenile and largemouth bass compare to the mangrove rivulus? Because morphologically they seem a little bit different.
Yeah, that's a great question.
The second one is, do you think they could jump on land if you ablated them off themselves?
Okay, so the first one, so it would be really nice to look at more similarly related and, more importantly, more morphologically similar fish. I've looked at one of their sister species, marmoratus, which is also a self-fertilizing hermaphrodite, and they also do the tail twisting. And you're right, the surface area is different, the body profile is different enough that it's probably affecting how they distribute their ground reaction forces. The other one, you can ablate their moth neuron and they will still perform an aquatic escape response. We haven't tried that yet terrestrially, but I have a feeling they probably would perform equally as well. So, Dr. Silver.
So you showed at the beginning that they would jump out of the water to get food.
Mm-hmm.
But did you ever record and do the kinematics for that? Is it the same as is it the same as the escape response? So we'll jump out of the water to escape predators.
Mm-hmm.
We'll jump out of the water to feed. Is that the same response? Same movement?
We see the same S-bend, and the duration was the same.
Because the escape response should be automatic.
Yes.
But feeding should be...
Yeah, it's a different sensory system.
Or it's a different
and
brain...
Yeah. So based on the duration that we recorded, it was the same, but I didn't look at that with the goggles of an escape behavior, because we only saw that pounce when it was pursuing a prey item. But the duration was the same, so I wouldn't call it an escape response, because it was all voluntary. So it was never, we never elicited any stimulus to generate those launches or pounces. It was always active.
So in the kiddie pool, when you were touching them, that was...
Oh, that? Sorry, I thought you meant in the transition. So for the kiddie pool?
Yeah, well, in the transition, they get escape responses, right?
Yeah.
And you put something in there to scare them.
When they were transitioning from water to land? Never. It was all voluntary.
Okay, but that's because there was food.
Yes, we conditioned them to pursue a cricket on land.
Right.
So.
But you never, in that setup, you never...
Correct. We never, ever generated an escape.
Okay, that's my question.
Did you ever see if there was a bias for one side or the other whenever they started their...
So if there's left or right-handedness?
Left or right-handedness.
Yeah, not statistically different. Because they would perform equally depending if they're on their left side or right side. Yeah, it's a good question, and it was really kind of fun to look to see if there was a particular side that they, I don't want to use the word prefer, but if they would perform more effectively, if it would be a greater performance generated, and there weren't any differences. Yeah.
I was confused by your answer about the mouth neurons. So how can they ablated mouth neurons still allow for C-start? I thought that was the yes or no, go, don't go. I thought that's where it was generated. So
it's not just those two neurons. There's been a lot of studies, and John could share some papers. I can share some papers. So it's a series of neurons in that network escape, and they can still perform those behaviors.
But not required. So what's your prediction then, based on what you've seen in the differences between the terrestrial and aquatic escape responses? Is there different coordination of the same neurons, or is it a different neuron set?
Yeah, I don't know, which would be fun to explore. Yeah, I wanted to take you guys down the pathway of neural circuitry type studies, but they don't exist for these fish yet, and yeah, we don't know. Kathleen?
So Mike asked about size class distributions. I guess the habitat is very different for the bass and the rivulus as well. So I was just wondering very simply, why did you compare those two?
That's a great question. So it's a little ridiculous. So this whole study is part of a greater jumping fish study. So looking at other fishes that are within the same order and then very distantly related fish. So we had the opportunity to get some bass very quickly, and we had them that were size matched, so we figured we might as well compare them. So that led to, well, we have this quasi-amphibious teleost, let's compare it to an exclusively aquatic teleost. So in a perfect world, it'd be great to have something that fits this particular profile and see what that does, especially something that looks like this that is exclusively aquatic, and see how that performs on land. So that is a great question of how we sort of serendipitously worked with the bass. But in terms of substrate, one of the things we'd like to do in the future is manipulate the substrate type, because we know that they will jump differently off certain grain sizes, so rugosity is extremely important in these fish, especially because they're so small. They're about the size of your thumb, if I didn't say. They're usually 4 to 5 centimeters in total length. And sometimes they'll be on leaf litter, sometimes they'll be on other branches, and it's hard to make purchase with some type of flat ground, which of course is going to affect your trajectory once you're in the air, or getting to that point. So hopefully that was yay bass.
I think it's a little silly, but I was struck by how long they can spend on land. You mentioned several months. Is their posture on land with that tail flattened out, or are they just sort of slumping there, and that's unique for for when they jump?
So they'll jump from both their side and in the prone position, and you'll see them at rest in both. So I've only ever seen 2 fish.
I just wonder how they stand around in the hollow logs or the crab holes.
Yeah. So the logs are hollowed out, but it's just a gallery based on termites and beetles and things like that, and there's really not much room at all to move. But they find their way in there, and that video, that grainy one of the first videos to capture these fish, there were about 30, 35 fish in this tiny little log. So how are they doing it? They're probably not moving around too much, but as long as they can find their way back out to eat some nutrients, find nutrients somewhere, and of course not desiccate, that's going to be their limiting factor. Any others?
All right, let's thank Ben again.